Welcome to this presentation on magnetic particles for protein separation and purification, brought to you by CD Bioparticles. Let's dive into the fascinating world of protein isolation and purification using magnetic nanoparticles. Proteins are essential biomolecules composed of amino acids, which contain hydrogen, carbon, oxygen, nitrogen, or sulfur. They play a critical role in the growth and function of organisms, from brain activity to muscle contraction. Beyond their biological functions, proteins are also used as diagnostic agents for treating diseases like cancer and neurological disorders. This makes the isolation of proteins from natural sources highly significant in biomedical applications. Traditional methods such as chromatography, mechanical disruption, chemical approaches, and electrophoresis have been used for protein extraction. However, these methods often struggle to achieve high purity for specific proteins. Therefore, there is a growing need for novel approaches to improve protein extraction and purification. Nanoparticles, particularly those ranging from 10 to 100 nanometers in size, or even up to 1,000 nanometers in some cases, are gaining significant attention in biomedical research. Their small size, unique morphology, high surface-to-volume ratio, and surface charge-mediated properties make them ideal for various applications. Among these, nano-sized magnetic particles have emerged as a promising tool for protein extraction and purification. Iron and iron-based nanoparticles have been widely employed in protein extraction processes, offering the added benefit of reusability through demagnetization. Iron oxide nanoparticles, such as magnetite and maghemite, have shown particularly promising results in protein separation and purification. Additionally, various metals and metal oxides can be transformed into ferro or superparamagnetic materials when reduced to the nanoscale. This chapter will explore the different magnetic nanoparticles that are beneficial for biomolecular protein separation. Let's now move to Chapter 1, where we will discuss magnetic nanoparticles and their applications. Magnetic nanoparticles, or MNPs, have found applications in a wide range of fields, including drug delivery, tissue engineering, magnetic resonance imaging, cancer therapy, clinical diagnosis, and even information storage. Their magnetic properties, combined with high yield, purity, and specificity, make them particularly suitable for biological applications. The interaction between proteins and nanomaterials is a key factor in protein separation. The formation of the protein corona complex occurs due to rapid interactions between the nanomaterial surface and the surrounding biological media. Nanoparticles with a large surface area and high surface free energy tend to form stronger interactions with biological macromolecules like proteins. In fact, the formation of protein corona in a mixture containing various proteins is a competitive process, leading to the selective purification and separation of individual proteins. Scientists can adapt this process to purify high-abundance species that rapidly bind to nanostructures. There are two types of protein corona: hard corona with high affinity values and soft corona with low affinity values. Factors such as surface properties, functionalization, size, and target protein concentration can influence the thickness of the protein corona formed on nanostructures. Now, let's move to Chapter 2, where we will explore the methods of formulation for magnetic nanoparticles. One common method for synthesizing magnetic nanoparticles is coprecipitation. This approach involves introducing a base to an aqueous solution of Fe2+/Fe3+ salts at ambient temperature under an inert atmosphere. Factors such as the type of salts used, the Fe2+/Fe3+ ratio, reaction temperature, pH, and ionic strength significantly influence the composition and morphology of the resulting nanoparticles. Controlled oxidation can also be used to synthesize maghemite, which is stable in both acidic and alkaline conditions. Another method is thermal decomposition, which involves breaking down organometallic compounds in high-boiling organic solvents containing surfactants. Common precursors include metal acetylacetonates, carbonyls, and cupferrinates. Surfactants like oleic acid and hexadecylamine are often used to stabilize the nanoparticles. This method can produce monodispersed nano-sized crystals with precise control over size and morphology. The sol-gel method relies on the hydrolysis and condensation of colloids to form a metal oxide network. The size of the particles produced depends on factors such as the hydrolysis rate, solvent content, pH, and reaction temperature. This method allows for the production of highly uniform nanoparticles. Finally, the microemulsion method involves creating a thermodynamically stable dispersion of two immiscible liquids, stabilized by surfactants. By adding solvents like acetone or ethanol, the nanoparticles can be precipitated and collected. This method is particularly useful for fabricating magnetic nanoparticles with controlled sizes and properties. Now, let's move to Chapter 3, where we will discuss magnetic particles for protein separation. One of the key applications of magnetic nanoparticles is the isolation of biotinylated proteins. Streptavidin, purified from the bacterium Streptomyces avidinii, has an exceptionally high affinity for biotin. Magnetic particles coated with streptavidin are widely used in molecular biology to enrich and separate biotinylated proteins, such as antibodies. These particles improve the throughput and accuracy of immunoassays while reducing nonspecific binding. Another application is the isolation of tagged recombinant proteins. Tags such as FLAG, His, or GST are used to purify recombinant proteins using magnetic particles coupled with specific antibodies or ligands. This method allows for mild purification and subsequent analysis of recombinant proteins, even from complex biological samples. Magnetic particles conjugated with protein A or protein G can simplify the enrichment of antibodies in samples. These bacterial proteins have high affinities for the Fc region of IgG antibodies, allowing for efficient purification. The elution of antibodies can be achieved using acidic buffers or urea, making this technique highly versatile for protein-protein interaction studies. Phosphorylation is a key post-translational modification that regulates many cellular processes. Magnetic particles loaded with metal ions like Fe3+, Ni2+, or TiO2 are increasingly used for the specific separation of phosphorylated peptides and proteins. This approach is particularly valuable in proteomics, where phosphorylation plays a critical role in signaling pathways. Protein glycosylation is another important post-translational modification, often associated with diseases. Magnetic particles coupled with lectins or oxidized glycoproteins are used to enrich and separate glycosylated peptides and proteins. This is crucial for biomedical and biopharmaceutical research, where glycosylation analysis is essential for understanding disease mechanisms. Now, let's move to Chapter 4, where we'll explore the future perspectives of magnetic nanoparticles in protein separation and purification. While magnetic nanoparticles have shown great promise, there are still challenges to overcome. For instance, iron oxide nanoparticles must be functionalized with biomolecules to interact with specific proteins. However, this functionalization can increase their hydrodynamic size, affecting their efficiency. Future research should focus on improving monodispersity and stability, possibly through the use of capping agents. One promising direction is the development of nanocomposites, where magnetic nanoparticles are blended with other metallic or carbon-based materials. These nanocomposites can enable one-step protein isolation. Additionally, nanoformulations like dendrimers or liposomes can release magnetic particles upon exposure to a specific magnetic field, allowing for targeted protein separation. Magnetic nanoparticles are also being explored for advanced disease detection and diagnosis due to their high detection limits. However, issues like agglomeration in body fluids and size increases during large-scale synthesis need to be addressed. The development of nanocomposites and nanoformulations will be key to improving their performance in these applications. This presentation is based on research from several studies, including works by Ivazadeh Kihan et al., Franz Reb et al., Onge et al., Zhao et al., and Vidarathanam et al. These studies highlight the advancements and potential of magnetic nanoparticles in protein separation and purification. Thanks for watching. For more information, please visit www.cd-bioparticles.com.